Okay, everyone, so it seems it's a long time. Yeah, and since the last lecture, and I'm okay. Now I start to share a screen. So today we're going to talking about the third part. It's the optogenetics in biological system. Today we're going to talking about the GEVIs. That's the genetical encoding voltage indicators. So generally cell optogenetics toolbox contains the two main compartments. So one is including that those families members about the tools to interfere with the cellular process, like the light control or light activate, like a biological process, like those rhodopsin, those channels, and light activate like rhodopsin linked, like adenylate cyclase or PDE, and all that is to interfere with the cellular process, and that's like some biological mechanisms that's being controlled by light. But there are also tools that using the light to be a signal to reflect or to show the cellular events. So we already know that those famous fluorescent protein like GFP, it gives the fluorescent the light, then it can indicate some specific proteins location, that's a function. And those tools to monitor the cellular events was also be considered as optogenetics toolbox within this super big family, but there's like a two part, like the first part like a taurodopsy, and second part, and that's those indicators. So in this chapter, we're going to talking about the voltage indicator. That's the voltage indicator, the GEVIs. Is at this stage, and actually they were more and like and engineered by scientists. It's not like those like channelrhodopsins or or the panrhodopsin, those proton pumps or chloride pump that are directly isolated from the microbiome, and then like to express here generously into like a mammal like cells or human like those cells like HPK cells. Um, so this is so in this part, in this lecture, we're mostly talking about some that is a genetically engineered proteins. So because today's we focused with the voltage indicator, so it requires a VSD, that's the voltage sensitive domain. And because the signal need to be like concentrate or need to be amplified, so and at this area, this FRET, the fluorescent resonant energy transfer, um, is this, we can say that a physical or chemical reaction or phenomenon was also be implied in their like experimental designs. So as usual, we're linked to some recent news. I find that a very deep sea, a latent fish was being discovered to on a beach. I think it's on the California beach, but I can go to see it. But it's very unusual. But it's just a really deep sea creature, and it's been appeared on a beach. And so another part that I would like to give an advertisement for specific projects, that's ocean project, and we can see that I already buy one of this turtle necklace to support this ocean project. From all these previous lectures, I have already discussed a lot of those like molecular device. All those proteins are actually coming from like the lake, from the river, from the coral beds, from the sea, and those can like really help our human beings to do to treat our human beings our disease, and for diagnosis, and for scientific research to satisfy our curiosity. I think these are all from the nature. So one thing we need put effort like to understand the get like get some like benefits from the nature, from the world, but also we need to like give back and at least protect those nature world on our earth. So in this lecture, I would like to talk about a little bit of this ocean project. Okay, so why I mentioned the Latin fish in previous slide, because in one of foreign one molecule that is being generated in 2017 by Gong's lab, etc. And they like combine in the deep tissue, there's no light resource, and there's also no external like LED light or like the fibers that provide the light resource, but they're very clever to provide another fluorescent protein to provide the light signal, then to activate or give like the light and energy to another protein. So that's what I'm thinking about. That is so close to the Latin fish, because there's in the deep position of the tissue. It's really like in the deep sea. There's no light resource, and there's no light penetrate from the skin, from all the tissues, and that protein that's being generated, it's quite like those Latin fish. So that's why I put the Hughes news in the beginning of my lecture. So the GEVI, the genetically encoded voltage indicator, it's a temporal resolution consists with the millisecond time scale of synaptic communication. And comparing to other like chemical or organic like voltage sensitive dye, it can identify the cell class identity, like expressed only in like the pyramid cell, and leave all other ganglion cells or local interneurons as no signals. So that will reduce the background noise and to isolate the signal that the scientists will be interested in. The calcium image provide little or no data of hyperization and subspatial depolarization importance because the GEVIs, some of GEVIs, if they're like embedded in the membrane, especially for those subgroups, they can sense this substratal depolarize rises, and this we'll be talking about later. So here is the big picture of a different subtype of these GEVIs. So some link some fluorescent protein then with a voltage sensor domain. So for this group, for A group, so it's generally it's coming from potassium 2.1. That's those 4 transmembrane domains that's for sensing the voltage change. But another group is the opsin-based GEVIs. They are using like rhodopsin-based logo to be the voltage sensor, like Arch, and that will be in the following lectures, and I've been focused on this. And other like combine those like these proteins and also with the dyes. Link some chemicals that's embedded into the membrane, link with the protein. So here the protein is like an anchor to target it to specific cells, and the sense of voltage change were located in those chemicals at the binding. So another group is the bioluminescence group. So in this group, so generally say, especially for this Lotus V, it's kind of like using the Lucifer rest coming from the fireflies that convert a chemical substrate, then provide the light. Then if there's like a voltage change and there's a conformation change, and it will cause these 2 parts that are close enough, then this energy transfer will happen, and the fluorescent, the light intensity of another parts, and it will be changed dramatically. So in this way, to indicate that there's a fluorescent change, because if there's a single only this molecules being attached, this fluorescent change will not be so much as being amplified by another specific fluorescent proteins. So that's the base of their experiment. So first, I'm going to talking about a little bit about the fluorescent resonance energy transfer. So generally say that it depends on the distance of the 2 different proteins. So if those 2 molecules were close enough, then the energy can be transferred from from the donor to the acceptors. But if the distance will be changed, like become longer, then the have a long range, then this like this energy transfer will be failed. So this phenomenon is a distance dependent coupling energy, like intermolecular, where it is, it's a intermolecular coupling. So it's really, so from the donor to acceptor, it's really determined by the distance of these 2 different molecules. So this is an image and shows now there's that if there's a more close of this protein, and then there will be an overlap between this an emission light wavelengths and with like excitation wavelengths of another protein of the acceptor. So if there's an overlap, so if the distance will be close enough, then the frets will happen. So typically this distance was 3 to 6 nanometers. So there are many different like applications for a two parts separated, and if there's a conformation change or there's some ligand like bind together, then this threat can happen. Or like the receptors then binding to specific ligand or substrate, then the two ends of the same proteins may approach together, and it can also be detected by this threat. And another one is like this binding of the two different parts, or another is that before there's a signal, but if there's an enzyme and cause the cleavage and this threat disappears, that means that there's protein like proteinase reactions will be happening here. So okay, now let's move on to see the first part of these GEVIs. First, using the single fluorescent proteins. So generally like there's three groups. So the first group that's those voltage sensors, the four transmembrane coming from potassium channels that are linked to this, because it's not functional, like modified fluorescent protein. Some designs that put the fluorescent protein at the intracellular, and some designs put this fluorescent protein extracellular. So for the voltage-gated channels like potassium channels, it has four subunit forms a tetramer, and each subunit coming like this. So there's a four transmembrane domain that is a voltage the sensed domain, and the two transmembrane domain is forms as an ion selective like a pore that's for the potassium to flux into the cell or get out of the cells. So this four transmembrane domain is for the voltage sensor, and it can be cut and linked to other like fluorescent protein or something else that can sense the voltage change, that they change the fluorescent light of the fluorescent protein, and that's the basic design of the CIVSPs. And these GEVIs were developed in 2005. So like for another group, it's using the frets, is that like, so at the resting membrane potential, so there's a far, so these two are far away. So on those excitation light, it will give out like a blue light. But after depolarized, they make this two part close enough. So if there's a fret happening, so this blue light is gone, and the emission is coming from another proteins, and the frets happens, and the yellow light will be coming out. So this use the frets, fluorescent proteins are like the mermaid, the VSFPs, and the chimeric butterflies, and then those are using the frets and linked to a four transmembrane domain of the potassium channels. So like this is a show as a 2007, like Dimitrov is a suture. There's a report show that the CIVSD is using this concept that linked to a pair of acceptor and donors added to the potassium channel voltage change the voltage sensor domain. So in 2008, it's another one that's like called you mean Kinoko, and that's called this one MUKG that linked to MKO Chi, and this orange fluorescent proteins are from a color, and they say that there's a orange light fluorescent is much stronger on comparing to the previous fluorescent protein using. So here, like enhance the signals here. So for the PSD butterflies, like kind of like making a chimera that mixes how to say it's this one, this Yomi Kinoko, the griffin proteins from the soft coral. So this from soft coral, and this is not from potassium channels. Mixed with this one and with this one, so we can see that they're just like cut to keep like a part of the transmembrane domain from this coral like proteins, and another keep another part that is coming from the potassium channels. Then they just keep like N terminal and another is a C terminal, kind of like that. It's like quite kind of similar to the C1 C2 and the chimeras that to see that the results if it becomes better because there's many aspects that need to consider about, because like a membrane targeting, expression level and the toxicity, and there's many like factors will be affect to determine to choose which the combinations will be best, more suitable for a voltage sensor. So cameras, and let's choose one is this is C5, this one. So just keep a little bit of the potassium and another potassium channels voltage sensor domain, and another constructor choosing the C25. That's like keep a majority of the potassium channels. So like compared to like the C25, the blue one, and another one is with FP and the C5. And it all forms like the sensor at functional at a different like the membrane potential and the current that both the C25 and the C5 will be bettered than another like the positive controls. And the time constants is also shows like a better response. However, well, we can see that that's the difference change like only like 3% of resonant change at the different voltage levels, but actually this is quite small. But comparing to other like voltage indicators, that those range will be among like 10%, 20%. So it's all that in that range. So of course we can say that the voltage sensor still have a long way to go to show that there's a voltage change. But in another way, when you say that, because like the action potential, and then the total voltage will change like -70 to like a +20. That's more than 100 millivolt change during an action potential. So in that way, when you say that maybe this like a 3%, 5% like difference change, or could be enough. So it depends. Okay, so another is like the single fluorescent protein that's not using for the frets. Okay, so one example is the arclight. It expresses fluorescent protein in level 1 and level 5. So I guess maybe those level 1, maybe it's a dendrite process. Like we cannot say that very clearly, but so when using these indicators for experiment, so here's a pretty cool. We show that like open the scale of the mouse, then directly to observe the cortex like the voltage change and activities on the brain. That's pretty cool. Another is the VSFPs, and like the wife truncates that they only keeps CFPs. Okay, another like developed in 2009 in Knöpfel's lab. First author Guntem and is also this chapters also. And the development, I think, is quite interesting because it's a circular pre-mutant fluorescent protein. So their design is like the fluorescent protein like from the 1 to 228, and they make dramatically change that the encoding to like from the 144, like insert the end um in the how to say in the center of the sequence, and also including the first amino acid. So before we can see that it's like a linear, but it just like folding together, then to forms GFP's a bucket structure. But here, because they link the first and the last amino acid together to form, so it change the beginning and the end, and then they change the whole like the amino acid sequence to form a circular permutant fluorescent protein. I think that's pretty cool. Not sure if this video should still show this. I really like this design. I think this is very clever. Because in this designs, the fluorescent protein is still like a functional, but there's some property change for that. It may change, so the change like the linked to other proteins will be at the different positions for the mutant one comparing to the original one. Okay, so here is shows the result. So the change rate will be like 1%. And in another lab, like quite recently, 4 years ago, and this is the flicker R, and it link like this voltage, the transmembrane domain, and with this also circular permutant fluorescent protein CPM apple, and they do direct evolution that's been estimated with a yellow light to isolate those fluorescent proteins that can. Okay, first I need to confirm that it's and it can like some difference like a sense to the voltage change. So to isolate it, then to do some that have very bright fluorescent protein, and to do the evolution um iso isolations for choose the best candidate of these GEVIs. I think that's a pretty cool. And you see they can quite catch the voltage change, and they show that this flicker R is better than arclight. So the third one is the SAP1, and the difference that this fluorescent protein was being located in the extracellular part. So the rest of them are located in the intracellular part. So those extracellular parts, but actually, so that shows that there's more bright signals. After depolarization, so these signals will be gone. So to indicate the fluorescent, indicate the voltage change. Yeah, to indicate the fluorescent voltage change, that after like a more depolarized of the intracellular membrane potential, actually fluorescent is going to negative decrease. So then in the second generations, it's also that the fluorescent is located outside SP3, and for this SP3, they use an AV to like transfect the neurons and can recording like the brain neurons activity during an awake and moving like animal models. So another group is like combined with rhodopsins, like acherons and MQ, citrus. So this like they linked the fluorescent protein was not the voltage sensor domain of other like voltage gated channels or the UKG, those the coral, isolated from coral, it's linked to rhodopsins. For example, the acherons. Actually, this we're talking about these arterioles in chapter 6. At that time, like I'm not quite clear on what's the mechanisms of this arteriole 2, but now I think I'm quite clear. So one thing that so for arterioles and endogenous, it have some colors at the pH at the neutral pH it shows like a red color, but in a very kind like in a environment it shows a yellow light. But in this paper also describe them like it's no color. So anyway, so at the different pH conditions, the color of these electrons were different. And that's because when the concentration of a proton uh, um, were like quite different in between these two conditions, and that were determined the location of the protons within these rhodopsins, and then they will cause the color or absorptions on light wavelengths will be changed. So the protons location will not only be determined by the pH difference between comparing to the different side of the membrane. It can also be affected by the membrane potential for sure. If there's a more negative membrane potential, then the protons will be preferred to be located on like out of the center of this rhodopsins. So that's why so it's not really determined like with there's a relationship between the pH and the membrane potential, but like either the pH change or the membrane potential change, then both reasons can cause the location change of the proton within the rhodopsins, and we can also change the absorption or emission light wavelengths of these rhodopsins. So it shows up here that the signals were quite different between different pH. So this is from the RG proton pump. This AR actually is AR3. And I think there's some change. Let me see. Will keep it the indentation discard. So I would like to indicate that it's here. It's 92 in this location. Is quite critical for the proton pump because it's required that if like using it as a voltage indicator, we cannot get this light activate the photon current to be involved in the cellular process. So we need to like do a mutation at this position, then to make it like make it less than no like the proton current that flux through this pump. So there was only like a conformation change and the color change of this rhodopsin, and we don't want any like protons like a change the intracellular pH of the targeting the cells. So this is also from the chapter 6, and here is to show that for endogenous for this rhodopsins, it has different colors. That's due to the interaction of the retinols with the proteins will be changed. So arterioles were actually the best. That the neutral pH, basic arterioles are pink, but at higher pH, the protein turns yellow. But there's some problems. The cells shows a 70% photo beige over continuous 10 minutes exposure and retain normal for during these intervals. And the first member of this group displayed in fluorescent has required high station intensity and a high specific optical image setup to image. A second potential cavity of the probe is a residue for the current. So although we do a single mutations to block this for the current, but there's still some residue for the current left that may cause that's unwanted effect of this current of this voltage indicators. So another is also do a direct mutation to isolate it, and that's the arch MO2, that's quest R1. It's also do direct evaluation and to isolate a voltage sensor. And we see that this works on HEK cells and also on the pyramid neurons of the hippocampus, and it kind of the brain size. It also works could nicely expression on the cell membranes. Then the next part is love rights of for this threat option GEVIs, there's a quite similar with the previous discussed about those voltage sensor that are linked to a linked fluorescent protein. Then use the threads. So the first part is they make you citrins and transmembrane part, and the voltage sensor is the MAC. And another group is the chemi-genetic hybrid GEVIs. They combined with the chemicals and also with the proteins. Different designs. And first, I'm going to talking about the STVI ones. STVI one is more like this one. Let me see. Yes. So it's a very high, how to say, it's a very large chemical molecule, and the voltage sensor is near red, so it has the color of a red color, and this is being applied before experiment, and it can be docked with a protein tag, and this protein will also link a membrane targeting like the GPI anchors. So structural property compares two different components. The first one are targeting indicator to different cell types, that's the protein part, while the second part is a fluorescent dyes that need to be delivered to the tissue, commonly via intravenous injections to deliver to the tissue. So before this chemical dyes is being sensor light, and it can inserted into the membrane, and it can sensor light and can check the voltage change quite good. But we cannot make sure that it's targeted to those cell types we want. So the science do those light then linked to binding to a shape that's the protein, and the protein will have an anchor and located to the membrane, and the protein will be expressed in specific cell type. So in this way, you can make sure that this chemical dyes that will only be binding to specific proteins that on specific cell type. and this design is a STVI. So for another opsin dye frets, such as Levotron and the Postron, is also developed in the same group. So generally, the idea is that the change so previous other let's crosstalk or molecule dipole coupling interaction between fluorescent protein and the opsins, and here the donors will be changed to a chemical dye, and this chemical dye will be binding to a halo tag, and this is a protein pocket that can bind into these chemical dyes. So after depolarization, there's some fret reactions between these two parts. So the fluorescent were not coming from this. It's a GF dye, and it will be this energy will be converted to the opsins and it will cause the fluorescent change. So this fluorescent was between this reaction and interaction transfer or between this transmembrane domain, and that's the rhodopsin, and between and another part that's the donor is the JF chemical dye. And the JF is a general fluor dye, and it also covers a wide range of visible spectrum. So it can be determined by what's the excitation wavelength to choose which dyes to evolve in this voltage sensor indicator. So another is the voltage spike that's linked. So before it's only this molecule dyes, but the current the voltage spike is like using a linker and linked to tag into linked to proteins. That's a design is very close to the virtual. So another part is the bioluminescent GEVIs. So for the later developed the Lucifer bust is a Lucifer it can provide intravenous provide the substrate all of this Lucifer rust and can change it can do chemical reactions and then it can generate a light energy then transferred to fluorescent proteins, and that's the bioluminescence. And for amber is another type. These two parts will be linked together, but the protein action and conformation change was between these two between the flavin reductase and with this complex. So I'm talking about so at the beginning of this lecture, we're talking about the Latin fish, and so here I'm thinking that this bioluminescent GEVIs, it's just like a Latin fish that and in the deep tissue there's no light, so you see this luciferase from the fireflies, and it can provide the light energy that can activate the fluorescent protein, and these interactions will be determined by the membrane potential change and by the conformation of the transmembrane domain to make sure that these two parts are close together. And it's really so when here it's give the trigger or lighter, and the YP were like a bonfire that provide a most intense light signals for the device to capture of the membrane change. So that's the same that's like the hyperization that's too far away and only very close they come together, and then this light signal can be converted and amplified by these fluorescent proteins. Gamma voltage free from excitation light and the migrant artifact will get to a local concentration change by luminous substrate by ratio process. So for all this, because like in the biological experiment, we will use a lot of light, like some like need to like GFPs green light that to identify specific cell type, or some transgenic mice that use the fluorescent protein as fusion proteins indicators. So for the voltage indicators, that's very preferred at using this red and near infrared light signals to be the voltage indicator. And also, this near infrared can diminish light scatter in the tissue because infrared light, like longer wavelengths, can penetrate tissue better and decrease the tissue autofluorescence and reduce absorption of fluorescent excitation emission light, but also by hemoglobins and like there's a dynamics change. And also another issue is that's important for this experiment is that the moving from image in brain size to live animals will increase the complications of this experiment. And we also know that like for subrotational voltage change, the fluorescent change will be only like 3% or 5%. And if there's the breeze or the different focus out of the focus because due to the animals is breathing or like the movement, and it will all cause like the signals change because it's out of focus. But this is not due to the neural activity change. So this will be all be considered to improve all this method. Voltage signal for individual are more susceptible to contaminated by voltage signal of neighbor cells and process image on the same pixel of detections. So there's a two approach, like to improve the experiment is like for limiting patient to the cell body, like using specific like Kv2.1 potassium channels, like the targeting signal peptide that can locate to the soma and express the cells like sparsely and intensity. So I need to like a very low concentration of the virus, but very strong promoters to promote the expression level of these proteins, like to make sure that the signals is shown enough to be captured. So for the image approaches, let's go like wide field epifluorescent image, and you will have a scatter light, a wide field with pattern illumination, for example, like the spot illumination, and deep tissue image use green lens and a fiber optic image, a deep image with a 2-photon excitation fluorescent. So like for two photon excitation, we can know that they can like use a longer wavelengths, and they can for better signal to noise ratio, and the fiber optic range, we know that is put all the signals with the optic fibers. So here I'm going to talking about this green lens. So green lens is like put the signals with this half ball lens and to focus the light signal coming from LED, then put the light resource to here. I can mount all this mini scope onto a live animal. And there's only one thing that I'm concerned is that why I don't use a rat, because rat is much larger than the mouse, and this headset for the mouse, I think it's really big for these animals. But this design, but comparing to other like the microscope, and this is a pretty good. It's a pretty much smaller, but comparing to the size of the animal, it's still super big. But I think it's still a good design. And later, I'm still thinking that maybe mounting on larger animals will be better and more applicable for these green lenses. But it's still pretty cool. Thinking about that is still a microscope. So there's many factors that I need to consider for GEVIs like kinetics, spectrum property, signal noise ratio, quantum yield, dynamic range, selectivity, interference with blood change, long-term stability, and cell toxicity, light toxicities, anything as quite important. So advantage of the GBIs, it provide valuable alternatives that can be genetically targeted to subset of cells that can identify the cell types. Light-dependent control of gene expression, protein degradation, neural differentiation, cell-to-cell interaction. That's okay. Yeah, I can record it with the light signals. And the disadvantage of GVS and low steps, we need to improve that, like a low bright field. Poor photo stability, slow kinetics, and low property to press membrane, although with the loss of membrane targeting uh, signals exhibit cellular toxicities. Okay, that's all. This is a reference and I'm talking about today. And so the court is the journey of a GBI development is only middle way. So there's a lot of ways to go, like to find and to develop more sensitive and more like better time resolutions for these voltage indicators. So there's a long way to go. Thank you.